Welcome to EPG Pathshala. In today's lecture, we'll look at sequence comparisons, particularly the two-sequence alignment as a first step in building up the comparisons of large numbers of sequences. Why do one compare sequences? Sequence comparisons provides a quantitative measure of evolutionary distance between two proteins, for example. Proteins with similar sequences are likely to have similar structures and in many cases very similar function, and that's something which you would like to know. And then sometimes people use it to infer, for example, gene regulation mechanisms, and there are lots of other reasons as well. Before we get into the business of protein sequence comparisons, it's quite important that we clarify certain terminology. Now, unfortunately, in this business of sequence comparisons are done by two groups of people. On one hand, you have the biologists who are comparing sequences of proteins or genes or whatever. On the other hand, there are computer scientists who are also comparing strings of characters for many other purposes, including that of biology. Unfortunately, the terminology used in computer science and in biology are sometimes different. So whatever in biology we call it a sequence, in computer science becomes a string or a word. If it is a subsequence in biology, it becomes a substring, contiguous subsequence. And if it's an alignment, it can be an exact matching or an inexact matching. Now, the reason this is important, because sometimes when you read the literature of sequence comparisons, the book or the paper might be written by a biologist using biological terms, or it might be written by a computer scientist using computer science terms. One has to be careful of what one understands from the words that are written. So, the first thing which we are going to talk about is aligning a pair of two sequences, maybe from DNAs or proteins, just two of them, and the methods for doing so. Now, why does one have to align sequences? This is quite useful, for example, when one constructs physical or genetic maps of the chromosomes. It's also quite useful in storing, retrieving, comparing protein and DNA sequences. It is imperative while doing homology modeling or while studying sequence structure and their interrelationships. So let's look at the different methods that are used. The simplest perhaps, and in my opinion, perhaps one of the best methods that there is something that we call the dot matrix class of methods. And then of course you have a large number of methods based on dynamic programming in computer science. Depending on the kind of alignment you're going to do, you might be using either the Needleman-Wunsch algorithm, you might be using the Smith and Waterman algorithm, you might be using programs like FASTA and BLAST and its various variations, etc. for alignment purpose. So let's look at the dot matrix plot. Conceptually, it's the simplest of them all. It's just like a two-dimensional graph. You have your sequence, one sequence in, say, the x-axis, another sequence along the y-axis. Now, if in a particular position, let's say the 10th position, the character in the sequence along the x-axis, which may be for DNA A, C, G or T, matches the particular character along the sequence along the y-axis, just put a dot in the position XY. So if there is a match, put a dot. If there's no match, don't do anything. Just do this along the entire length of the sequence. Here is an example of a dot matrix plot. Now, if two sequences are identical, let's say I have the Salmonella typhimurium sequence on the x-axis and the same sequence on the y-axis. Now, what would happen? I would get a dot along the diagonal of the protein. Now, in this particular case, what happens? On the x-axis I have Salmonella typhimurium, y-axis I have Salmonella typhi or Salmonella enterica. Now, most of the genome of these two are identical, and you can see this strong line along the diagonals. However, there are two cases where you see a line along the anti-diagonal. What does it mean? which means that in one of the two genomes, for example, that segment of the chromosome has inverted, and there are two such inversions, which is why these two breaks. Now, if there are, for example, no inversions or anything, there's just a mismatch, you'll just find there is a gap in that region of the line. And if there's a strong match, you'll get a straight line diagonals. Now, before going into the details of sequence alignment procedures, again, some terminology needs to be clarified. Now, the first thing is substitution. What does it mean? That you have a sequence letter for DNA, it may be A, C, G or T. So if it is A in one sequence, that A is replaced by T in the other sequence. If it's C in the first sequence, replaced by anything, A, G or T, not C of course. Then there can be an insertion. That is addition of one or two letters into the sequence of one of the sequences, and a corresponding position does not exist in the other. Or sometimes there is deletions. Now, if you have additions and deletions, the length of the two sequences are not the same. So suppose I have one protein which has 100 amino acids, and its homologous protein from another source has 150 amino acids, which means that compared to one, there are either 50 insertions or there are 50 deletions. Now, whether you're going to call it an insertion or you're going to call it a deletion depends upon what you consider as a reference. So if you delete something from somewhere, it also means the same thing as you've inserted something somewhere else. So in such cases, therefore, in view of such cases, we do not consider insertions and deletions are really different. In fact, there is a term called indel, which combines insertions and deletions in one. So essentially what we have, substitutions, where a character is replaced by another one, or you have indel, where you have one character in one place, it has been deleted in the other. Now you can call it there wasn't a character and something inserted, or there was a character and something has been deleted. Both are the same, so we will call it an indel. Along with substitutions and indels, these two things are the basic operations that one needs to do while doing a sequence alignment. We have a term for that, and that is called an edit operation. Okay. So let's define this thing called edit distance. That's the minimal number of edit operations that will convert one sequence into another. Now I'd like to give an example out here. As a child, and I was going to nursery class, my teacher would often call dictations. She would tell us words and we used to write it down on paper. And of course we would make spelling mistakes. Now, the number of spelling mistakes that we had made is the edit distance between what I had written and what my teacher had in mind. So let's define a sequence alignment now. So when comparing two sequences, you count these edit operations and you arrange the sequence in such a way that the set of edit operations which you're using has the maximum total score or the minimum weight. Now, doing these alignments, there are basic two types of them. One of them we call global alignment, the other is opposite of global, we'll call it the local alignment. So global alignment, defining it is simple. Given two sequences, say S and say T, what is the edit distance between them? For local alignment, given two sequences, S and T, what is the maximum similarity or minimum difference between a subsequence, not necessarily the entire sequence, just a subsequence of S and a subsequence of T? So the difference between the global and the local is that while the global alignment necessarily requires the entire sequences to be compared, in local alignment you are interested in finding the maximal similar part of one of the sequence which matches the other one. Now in so doing, when you are comparing two sequences and trying to maximize the matching between them, you might have to insert a gap or a space. A gap is a continuous run of spaces which are added into the sequences so as to maximize the score of the alignment. every gap represents an indel operation, isn't it? So sometimes it might be useful to penalize the number of gaps. So in such cases we use a gap penalty function which measures the cost of adding a gap, and it may be a constant, it may be a linear function of the length or the number of gaps, or sometimes it can even be a nonlinear function. So now let's look at the global alignment algorithm. This was first proposed by Needleman and Wunsch in 1970. Again, let me repeat the problem definition, that there are two sequences S starting from position S1 up to Sn, and a sequence T starting position T1 ending position Tm. Now, M and N are approximately the same, they can't be too different from each other. Our problem in global alignment is to find an optimal alignment, a matching which gives us the best score. In so doing, we just have to add gaps or spaces or indel operations in one or both of the sequences so as to have the maximal score. Now let sigma AB be the score of aligning one character, say a character A in S, which may include space, with a character B in T. Now A and B, they may be the same, they may be different, whatever. So the alignment score will be defined like this, that Vij is the alignment score up to aligning position 1 up to S1 up to Si and T1 up to Tj. Of course, I must be less than n, greater than 1, and j must be less than m, greater than 1. Now given that I have VAB, what we do in the Needleman-Wunsch algorithm, that for both the sequences we add a 0 position. So one of course has to look at what happens if the first character gets into the 0 position, that is before the first of the other one. So we have two base conditions. Vi0 is matching the first character or any character in S with nothing in V. Similarly, V0j, which is the score for matching any character in T with nothing in S. And once the base conditions are defined, one goes along the two sequences comparing the positions in all possible ways, and these are defined by a certain set of recurrence relation. Now as you move along, you can move along both sequence I and j in sequence S and sequence T. In that case, the first of the three equations given will be used. You may move along, say, sequence S, but not along sequence T. In that case, the second one. You may move along sequence T, but not along sequence S. In that case, the third one. So you compare the values obtained by doing all the three. Whichever one is the maximum, whichever one is the largest among the three, is the one which you use. So let's say the sequence S is along the vertical axis and sequence T is along the horizontal axis. Now if I take the character A and I start matching A in the vertical axis, the first character, and I start matching with whatever is along the horizontal axis, of course I give a score that for a mismatch I give a score of -1, for a match I give a score of +1. Now let us see what happens. Now when A goes with the 0th character, since it does not exist in T, I get a mismatch score -1. Then I see in the first character of sequence T is C. It does not match my first character A, right? So I get -1 again. And then in the second place, of course, it matches with A, I get a +1, and so on and so forth. I go on like this. Now once this table is filled up, I start from the bottom right corner, which in this particular case is the 6th position G in S and the 5th position in T, and then I look at which are the largest pathways. In this particular case there are 2 ways to go, right? And at each position I go and find out which are the largest backwards, and I go and move along until I reach the end of the sequence, the beginning of the sequence rather. Now As you can see out here that there are more than one path which connects the last position, the end of the sequence, to the first position, the beginning of the sequence, right? So in this particular case, there are more than one global alignment. But of course, when you are doing real life sequences, they are very large, you usually do not get too many detours. There is usually just one best global alignment. Now, one question which becomes very important in computational biology is about the time complexity of the algorithm of global alignment, which is to say that given a particular machine, how long will it take to construct a global alignment? Now, if I have, it depends of course on the length of the sequences being compared. So if the first sequence has length n and the second sequence has length m, then the time taken is proportional to the product of the lengths of the two sequences, that is of the order O and m, and the space required is about O n plus m, so it is about just the sum of the two lengths is required. If only VST is required, that is the final alignment score is required. If on the other hand you require the complete alignment, then even the space requirement is proportional to the product of the lengths of the two sequences. Now coming to local alignment, it's quite similar to global, saying that given two sequences S and T, but now we are interested in finding subsequences alpha in S and beta in T, whose optimal global alignment will be the best over all such pairs of subsequences that we can take. Which means to say that if there is a segment in sequence S and there is another segment in sequence T and they align best of all, find that. You don't have to consider the rest of the things. Now, no mention is made about what are the length of these segments, and they can be very small and they can maybe cover the entire sequences. You have to find out what is the best segment among all possible segments that there are and in both the sequences, and which pair aligns best. How do we do it? In case of long sequences of possibly unequal length, only short stretches of aligned sequences are really meaningful. Sometimes, as in eukaryotic genes, the gene sequences are made up of a concatenation of fairly large segments, the so-called domains or the exons, but in between there are lots of things which may not have an immediate meaning. So in this case, our introns, which are not really expressed. So when comparing such sequences, let's say I'm comparing the sequence of a gene from an eukaryote which has introns and also the homologous one from a prokaryote which doesn't have it. Now, while comparing this, I have to compare only with the segments which are translated or expressed in eukaryote with that of the prokaryote. The introns are not going to be useful. So the whole gene sequence is not really very useful in this case. I have to find out the subsequences which are best matching with the other test sequences, and this is where local alignment comes in. So differences in algorithm between the local and the global alignment are somewhat minor. While the scoring function in global alignment can be anything that you really wish, in local alignment there is a little bit restriction that if there is a match, the score must be positive. If there is a mismatch, the score must be preferably negative. Of course, you can't have both of them 0, then it wouldn't work. The recurrence relations, as we have seen for global alignment, are exactly the same before, excepting with the addition of a 0 out there. So you are taking the maximum of not only the 3 directions as you are moving and using the 3 equations, but if they are negative, you are putting a 0 there. What is the net result? As we can see in the next one, is in your table you have lots of zeros. So when you are trace backing your path, you suddenly stop at a point because you have zeros everywhere. So this, wherever you can trace a path, is a subsequence, and that and its corresponding partners in the x and the y axis, it gives you the best alignment. So that is all about local alignment here. Now one question about gaps. As one has discussed before, a gap is any maximal consecutive run of sequences in a single string in a given alignment. Gaps are very important in many cases, For example, when matching cDNA sequences with the corresponding genomic DNA, you will have a lot of gaps wherever there are introns. Long gaps, long series of gaps, if you see that in an alignment, or sometimes when there are no gaps in an alignment, both cases, can often be rather dangerous, and something is wrong there, and be careful when you see such things. For biological reasons, not necessarily computer science problems here, often one would like to have a control and the number of gaps and how they are adding. So what we do is that whenever too many gaps are being added, that is in the recurrence relations, when you are going at I, j minus 1 or j, I minus 1 on those directions, we add an extra penalty in those. So which can either sometimes favor or sometimes disfavor the addition of a gap. So the question is, how are you going to add your gaps? Someone may say, look, I really don't bother. I don't want too many gaps, but wherever they are, it's fine. So you use a constant gap weight model. Some would say that I don't want my first gap, I don't want a gap to come. But if it comes, I want all my gaps together. In that case, you use an affine gap weight model, put a high penalty on the first one, and then for each subsequent gap which is added at that position, you give a lower penalty. Now if you stop adding gaps and go on normal and you start another gap, again you start a high penalty. This particular model is generally the one most commonly used. In some cases, you would say, no, okay, I can start a gap, but I don't want them to be too long. In that case, as you start the gap, that's fine with a low penalty, but as you go on adding, your penalty actually starts increasing. And then lastly, you can have an arbitrary model, whatever you wish you can implement. So these various different gap penalty models, they serve to control the kind of alignment that you will get. So in summary then, what we have learned today is how to compare a pair of sequences. The simplest of them all, no mathematics, no nothing, is your dot matrix plot. Put them one along the x-axis of a graph paper, one along the y-axis, and if you have a match, you put a dot in. If the sequences are similar, what you get is a diagonal line. If they're close but not actually very similar, you still get a diagonal line but some holes and gaps in between. If they're not similar at all, they're quite different, you get a random mass of points. While this dot matrix model method is pretty good, it can be quite cumbersome. You can't be comparing hundreds of pairs of sequences. So you have computerized algorithms to do that. They come in two classes, global and local. The global algorithms compare the entire sequence, one sequence with the other. The local algorithms, on the other hand, find out the best segment of one sequence which matches another segment of the other sequence. The length of the segments are not a parameter. So they can be anything very small or anything big. Whatever matches best is given. And then finally, when you have a match, it's fine. You have a mismatch with something else in the corresponding position is fine. Sometimes you have insertions or deletions. How do you handle them? Do you want too many of them? Do you want too less? You can control them. These are called gap penalty models. Various models are there which has been discussed. Thank you.